In order to observe quantum biology, we need to build quantum instruments. The idea is to build instruments that can perform bona fide quantum measurements inside proteins, inside cells. So those are glorified microscopes that can look at quantum signatures inside proteins and check whether that said quantumness is being used for function. They're optical microscopes coupled to coils that produce either or both DC and AC magnetic fields, everything synchronized down to the nanosecond, because that's the time scales of relevance for the spin physics that we care about, that we think underlie magnetic field sensitivity, and with very good single photon cameras, because it's a feature of the proteins that we study, but it's also a feature of technological quantum systems that the amount of fluorescence, the amount of light that certain proteins emit is correlated with the state of the quantum state of particular electrons inside it. So fluorescence intensity is a quantum signature, is a quantum correlate, if you will. So what we would like to do in the future would be to check for how long quantumness is alive inside cells and check whether it's long enough for magnetic sensitivity to be achieved. For example, if birds truly are using the magnetic field of the Earth to navigate, the profound implication that electron spin superpositions need to be alive inside their cells for at least 750 nanoseconds. And the smaller the field, the longer the quantumness that the cells need to support. So this is the type of instruments that we are starting to develop. What quantum biology gives you is the predictive power on how to engineer those fields to do things. And in order to get there, the first thing is to make sure we can prove unambiguously that those things are being used for function inside cells. And the tadpole experiment was conceived to update the scientific consensus that those effects are both real and measurable, right? Even there are websites from US government that says, no, weak magnetic fields don't do anything to you because they're too small, they don't trigger a biological mechanism. And that's misleading, that's false. Tadpoles were chosen because of how common they are. There's nothing particular about tadpoles, but we would find similar effects in any other model organism that we picked.